Yeah, hello all. I'm doing this recording obviously to be blacked out, so it's just an oral recording as opposed to video. And the way I customarily do it, just as a reminder, is I am effectively looking at the test and giving you some questions. I just sent you an email about respiratories a while ago. We're talking about a better part of more than a month ago, and even though it should be included with this. You are looking at the anatomy on the take home. So let me just review with you the essentials of respiratory for this particular exam. E- Effectively, from a functional standpoint, the big problem with respiratory diseases are less acute, unless we're talking about COVID and really severe respiratory infections. That's more about micro to understand those. And really, it's long term abuse. And most of the time, it's smoking or environmental toxins, and sometimes heredity, and sometimes other kinds of damage. The big factor is vital capacity. Vital capacity is really the ability, the amount of capacity you have to move air in and out and exchange oxygen and carbon dioxide. And so, by classification, when you have a diminished vital capacity, it's a restrictive disease. What are the causes? Emphysema, perhaps most common of anything, and typically that's smoking. Lung cancer, other diseases like tuberculosis. Uh, those are all in the picture. The distinction is obstructive diseases, and that has to do with your ability to basically expel air, which implies that you have difficulty in bringing in air, and just be able to basically push out 80% of your vital capacity in a second. So sometimes people can't do that. And that's usually an obstruction. You could think of that with a lot of different things. And emphysema can overlap both of those, but that's the distinction. So restrictive usually implies it's what's in the exchange division. Remember, there's two anatomical divisions: the conducting, I beg your pardon, physiological areas that we classify. Conducting, bringing air in and out. That's all the way from your mouth and nose down to the terminal bronchioles. And at the end of the terminal bronchioles, the which is where that division ends, the exchange or respiratory division begins at the respiratory bronchioles. And you know respiratory bronchial because it opens up into alveoli. And so the restrictive diseases are more about that area because that's all about diffusion. Basically, the air is so close to the alveoli, it doesn't require any push or pull to get it there. It'll be sufficient that it will diffuse over. So, by definition, the structural end of the conducting division is the terminal bronchioles. Once you get to respiratory bronchioles, alveolar ducts, or the alveoli themselves, then it's not there. The other important feature of respiratory to understand is that, and I really played around with it quite a bit, and because all about when we put those little O2 sensors on your finger, it's the oxygen saturation curve. And the big thing to remember, it's an S curve. So it's not a straight line relationship. It's not a bell curve. It's not something geometric. It's not something a best fit that you're doing in statistics or anything along those lines. It is the big problem with that is that it stays high, and then once you start to lose oxygen saturation, it again, as oxygen goes off of those cells, the whole idea is they change shape and it allows more oxygen to go off. That's great when you're trying to bring oxygen in or oxygen out, oxygen to the cells or oxygen in your lungs. But it's awful bad under normal circumstances because you can quickly lose oxygen saturation. So that's really the warning. Once it starts to go below 90 on those little measurements that you can see on a fingertip, big trouble. Okay, starts after that, and particularly for people with respiratory problems. So, effectively, how does the air get in the first place into your lungs? It's all about negative pressure. Your lungs themselves, that sealed chamber on both sides, has a negative pressure slightly with regard to atmospheric pressure. And again, it's all about the relationship about pressure and volume. So when we inhale, effectively, that little bit of negative pressure increases because we're expanding the volumes of the lung in a closed chamber. So in a closed chamber, if you bring up the volume, pressure goes down. So it increases the amount of negative pressure. There's a little bit in there just to keep the lungs inflated, and that really inflates them quickly that are there. And that's the beauty of it. That little bit of negative pressure doesn't allow the breathing part, the alveoli, to collapse. They're very easy to reinflate. And And a lot of that has to do with a particular chemical, which is the surfactant. Surfactant is made by the type 2 cells in the alveoli. We start making it efficiently at about 26 weeks, between 24 and 26 weeks, roughly, of gestation, right about the juncture of the second and third trimesters. And, and effectively, now that we can make it in the laboratory, or harvest it in some other means, we can utilize it and spray it into preemies' lungs, and they have a much better chance of survival. That's surfactant. So that's really an important area. And those are really the source of substance of these roughly five multiple choice questions. I'm looking around here now to see if there's anything else that might be of significance to you. I think more of the anatomy is certainly going to be more reflected on the take home, knowing where the beg your pardon, the hyaloid cartilage, the thyroid cartilage, the Adam's apple, the cricoid cartilage, the tracheal rings, et cetera, et cetera, et cetera, all that kind of stuff. And I think that really will serve you well as we move forward into this. And then I will continue now dealing with the remainder of the test. So again, the whole idea, again, just to summarize that with respiratory, it's all about cartilage. Cartilage is, it's all hyaline cartilage, and the beauty of it is it basically keeps the system open. Sometimes they're rings with an opening, like the trachea in the back. Sometimes they're rings that give away to plates, like they do in the lung. And all of the above are significant because what they effectively accomplish is maintaining what we call patency. So the word patent, which is spelled like patent, like you applied for a patent to develop something, Patency, is mean keeping something open, a hollow tube that's kept open that doesn't collapse. So we utilize this, and there's a lot of different things to use it. They use elastic tissue and blood vessels, and here the cartilage is what performs that. So I thought those are significant features, which really brings us into the next area, which is digestive. So the big player with digestive has to do, and it really what kind of is the secret sauce, I think, is you have the uh, the peritoneum. You have, remember, it's two layers. You have parietal, which is pretty standard. It's what lines the wall, and it's the visceral one. And the visceral one is interesting because it kind of doubles over and folds and contains lymphatics and vessels and nerves and other structures, and we call that mesentery. And you remember when I sort of gave you the anecdote about what happens when you pull bowel out to examine and effectively push it back in, and it retains its normal shape. That's because those mesenteries are really what hold everything in place, whether it's the small intestine, the large intestine, the relationship of the stomach and the liver, because most of those are moving around fairly freely. They're designed that way, other than the couple of places that are what we call retroperitoneal. And retroperitoneal are really important. Okay. And retroperitoneal are like stability points, like the common bile duct. It's a stability point. Areas, where the pancreas, a stability point. Those are things that are sort of tacked down that sort of keep things relatively organized, but the mesentery do the rest. The lining is simple columnar. It's multifunctional. Okay. So there's a lot of things that are about that. So it has more than one function by and large. Okay. It is multifunctional in its very nature. It obviously, the secretions, the mucus itself, whether it's acidic or alkaline and tight junctions, basically allow it to perform certain amounts of function that are there. The cells are very, very interesting because they're so mitotic. Those are about the busiest cells we have as an adult in the gastrointestinal area because they replicate very rapidly, but they're damaged very easily because we're constantly passing substances over them. So just food and drink alone, because of the chemical nature and the mechanical nature, damage those cells. So I think that's an important area to consider. So typically the way I phrase those questions is all of these are true about the lining cells or the mucosal cells of the GI tract, except, and it might be a none of the above or the all of the above and all that type of stuff. The typical questions that I asked. Some specifics involve some of the cells. So in the stomach, the stomach has simple columnar and it has mucus that's basically alkaline in nature. So it can deal with the stomach acid. It has a lot of tight junctions. But the big player in those glands Remember, the glands have those three really interesting cells. They have parietal cells that make basically the acid and something called intrinsic factor, has the chief cells that make the precursor to pepsin, the first start of protein digestion, called pepsinogen. And lastly, they have those G cells or enteroendocrine cells that basically make gastrin, but also play a little bit of role in histamine and acetylcholine production that are there. So they're really important that are there. I made a big deal about how the food, the way I play it with this relationship I have with food, are about the local movements, and that's from the muscularis mucosa. Remember, that's that fourth layer, that interesting mucosal layer has these multifunctional mucosal cells that are the lining. They're so busy and they're absorbing and they're secreting and they're replicating. They have a sort of an elaborate layer underneath it called the lamina propria. You have lymphatics. You have areas that have impact on those interesting glandular structures in those gastric pits. And lastly, going deeper or going more away from the lumen or away from the center, you have that muscularis mucosa. So what it does is with stretch receptors, when food enters, it stretches the tube, and the muscles respond reflexively by contracting. So they basically hug the food, and all of that is about processing it, breaking it down, whether it's in the stomach, absorbing and secreting, whether it's in the small intestine, et cetera, et cetera. So there's a lot that has to do. So it's a really important. We call them local movements because of all the activity there in the mucosa. It has a much more elaborate submucosa than other structures because you have a significant number of blood vessels and nerves and lymphatics there to effectively receive the processed materials, secrete things, supervise those local movements. And remember, the nervous system is much more elaborate. They call it the gut brain because you have a nervous system that effectively works on its own from stretch and chemical and pressure receptors and osmotic receptors. So it doesn't require the typical overrides that a lot of other systems do vis-a-vis sympathetic and parasympathetic. Yeah, parasympathetic makes it busy, sympathetic slows it down, but they don't come into play unless you're in fight or flight. So there's really not a whole lot going on with it. Basically takes care of itself. And I told you the anecdote about me that they cut the vagus nerve, which is the source of that, going to the stomach, the gastric branch of the vagus nerve, because it limits the secretion of acid and those other chemicals in an effort to deal with my ulcer when I had those surgeries long ago. So, so that's important. You should know what happens regionally vis-a-vis digestion. So what happens, and a side note, there's going to be more than one of these because I don't think, as long as the dog is relatively quiet and we're not doing a bunch of stuff here, I can dictate. All the following things are kind of what happen in certain regions. So if we think of it, the mouth's busy. It's mechanical and it's chemical. You've got starch breaking down with amylase. You have lingual lipase. Okay, that's there. You have the chewing aspect of it. Okay, the only thing you have a lot of water in there, and water is Im- implicit in every one of these reactions. Every time we break down anything in the body, we use hydrolysis. Fancy word that means we're breaking it down with water, lysing it or splitting it up with the aid of water, and then all of these occur. The only thing that doesn't get started so effectively, not considering nucleotides and DNA and RNA, is proteins. So we don't start to work on proteins till the stomach. So the mouth is where we start starches. We don't touch them again till the small intestine. We start fats. We do those also in the stomach and in the small intestine. Proteins we don't start until the stomach. That's that pepsinogen, and all of these then are really impacted. The small intestine is sort of the area where everything really occurs, where we're breaking down everything to its component parts and finally absorbing it through that brush border and brush border enzymes, etc. So the distinction is the simple columnar, which is really the hallmark epithelia. It doesn't start till we get to the stomach. That's why when people have reflux disease and that acid coming up, why we call it heartburn, because it damages the lining cells, which are stratified squamous non-keratinizing. Okay. And at that cardiac sphincter, there's an amazingly sharp transition to simple columnar with the goblet cells and the mucus that's alkaline in nature to neutralize the acidity that's there and all the tight junctions. So it's really a transitional point between effectively a mechanical passageway where maybe obviously some activity is going on to a where we really begin to focus on the mechanical and then augment the chemical aspects as we get to the stomach, sort of preparing everything into that mush we call chyme, where little squirts of this highly acidic, highly concentrated stuff get squirted into the small intestine, very tiny, because it has to balance out the pH with bicarbonate, because that's an alkaline based environment to really function in as the arm of completing digestion and completing absorption. So, in contrast to the chief cells, the hydrochloric acid that's so prevalent in the stomach because it's the acidity that really is very, very protective because that will clobber any microorganisms by and large, with except for a few that are relatively acid, not a lot that are acid stable. We cover that a little bit. We talk about when we talk about GI diseases in micro. For instance, salmonella, stomach acid works great against Shigella, which causes dysentery. We see that a lot in war-torn and natural disaster areas. You don't need a whole lot of that. Same thing with cholera. So that's kind of those are underlying reasons why certain things are more problematic than others. So remember the hydrochloric acid and as well as intrinsic factor come from the parietal cells, and the chief cells are the pepsin and pepsinogen. And effectively, it's almost like when we make the pepsin, it is almost stimulating the making of more pepsins. It's one sort of like a mini feedback loop. And the big player when it comes to hormones when it's in the stomach is called gastrin. Sounds like gastric, which means stomach. So gastrin is the primary thing that's produced by those enteroendocrine cells, and it's really something to watch for. Anybody with stomach disorders, or like me with an ulcer, or even cancers, they look at that at gastrin levels. There's a very rare tumor that you don't have to know anything about this called ZE, which stands for Zollinger Ellison. And anybody who's had stomach problems, they inevitably work them up for that because any doctor can find one of those. They're so rare they can write a paper and get it published. So doctors look at things like that. These every so often you get these interesting oddballs that everybody, that's an easy case study to get published if that's what you're interested in doing. So eventually, as I commented on before, the chyme is the big player that's going into the small intestine. Chyle is something that's lymph, is the name we give it for chyle. Remember, we had something called the cisterna chyli, and that's where it's located. Oop, oop, I went too fast. As I look at these things, it's not necessarily so easy. I'm going to try to go through digestive now and then carry on with the other stuff. So, oh yeah, the other chemicals I mentioned them before that are important are histamine and acetylcholine. Acetylcholine from the parasympathetic nerves innervates and sort of energizes the gastrointestinal area, as does histamine. And as I commented before, the pills like Zantac, they've been around for a long time. A couple of they've pulled. One of they may have pulled the Zantac. I think Tagamet was one that they didn't pull with. I think that's cimetidine, and I have to look them up because I don't use them. But they are interesting because they're histamine blockers. They're basically antihistamines, except when we hear antihistamines, we always think allergies, but this is a different kind of histamine receptor. It's not one that's around. That's not antihistamines for respiratory area. These are antihistamines for elsewhere. Okay, and these antihistamines have to do with this histamine type 2 receptor. So basically, what happens with the food once the stomach gets done with it? It goes to the small intestine, in the duodenum. That's the next location. And it goes through that narrowing called the pyloric sphincter. Remember, from the esophagus to the stomach is the cardiac sphincter. Very tiny amounts, half teaspoon to a teaspoon. Very, very acidic, very pasty and concentrated. And all of those things play a role because when the small intestine gets the bolt of low pH, when it gets that highly concentrated osmolar particles, as well as to squirt just a little bit of stretch. Those are the three factors that get the small intestine busy. And I think that's an important area as well. So when it comes to the small intestine, okay, it's really where everything is absorbed. A little bit in the large intestine, we get some electrolytes, a little bit of water, a little bit of vitamins. The pH change is very, very significant. That's there because it basically goes from low to high. It goes from alkaline to acidities. This is the first time we really get a handle on fats. We've broken them down a little bit. Bile salts are coming in by way of the common bile duct. And I think those are all really important factors. The big chemical is with the one for the bile salts is cholecystokinin. That's one big player that energizes it. The other is called secretin, secretin. We'll stop it here and I'll pick it up in shortly thereafter and we'll finish digestive in this review.